to talk about, of course, the title speaks for itself, correlative microscopy, but actually this topic was, a lot of the content of this topic was spurred on by things that I experienced at previous BCA meetings, things that other people show, whether they were presentations, show-us your stuff. Kind of a little spark went off in my mind, and I bring a lot of that back home, which is probably why my provost continues to send me to such meetings. So I'm delighted to share this with you today. This is going to be sort of a hodgepodge of student images and student projects, but they're all basically towards the same end of visual data. And I'll work through this as we go. And so we're going to go back to low tech today after Jamie's very eloquent talk. I teach a lot of histology in my classes, whether it's embryology, whether it's gross anatomy, comparative anatomy. I get a mixture of students. Most of them are in the medical pipeline, a lot of them dental, veterinary. And I've always had a problem with students, particularly if they're undergraduates, and you ask them the question, where do you get fill in the blank? For example, where do you get a frog? The answer is not Carolina Biological Supply. There's a biology behind the animal. And I started to bring that into histology as well. If we're talking about, for example, cystic fibrosis, and we talk about thick, pasty mucus that wouldn't be moved by cilia, the description is one thing. Handing them a slide that might have come from Carolina Biological is another thing, where they actually have to use the microscope. But as I told you the other day when I showed the little Wi-Fi cam, students don't often use the microscope. So I've taken a different hands-on approach, and we begin with some sort of cadaver. Sometimes they're human tissue, most often they're cat. Biopsy the tissue, embed it in some sort of a polystyrene resin. Don't use paraffin anymore. And I have them break glass knives, make the sections, mount the tissues, stain them, photograph them, label them, enhance them, what have you, and build up their own image bank. So they're usually given some sort of an assignment. For example, compare muscle types in this cat cadaver, or for example, look at the different types of cartilage and characterize each one. So that's kind of where this started. And to make it easy, of course, like every other lab that's involved in undergraduates, we were using simply methylene blue stains for years, and that showed pretty much what methylene blue shows. It bonds to protein and lipid. And what isn't made of protein and lipid? You know, it's kind of a loaded question. Everything is. So it basically gave you cellular outline, nuclear membrane, and basically that was it. Obviously didn't show organelles, but more importantly, it didn't show even what I would consider fine structure that you should be able to see through the light microscope that would make a distinction. So this is an example of two student images that was on the muscle project. You know, let's make some slides of smooth skeletal and cardiac muscle. Striated muscle was easy. You can pick up striations. These are not the best sections nor slides, but again, they're beginning work. The striations are easy, and if you really squint and maybe take it on a prayer, you could count the number of nuclei, but it's pretty hard to tell the distinction between what is going to be skeletal muscle or cardiac muscle with a basic stain. This was done with an H&E stain, which is pretty common. There we go. So if you apply an appropriate stain to the same tissues, they jump out at you. And rather than just an anatomical description, then they can understand how it relates to physiology and function. So on your left would be, of course, skeletal muscle, and you can see where the nerve fibers literally jump out with a gold chloride stain, and you can actually see the little boutons where it interfaces with each of the myofibrils, and it really talks about muscular function, acetylcholine junctions, things like that. On the other hand, what is indicative of cardiac and only cardiac muscle are the little intercalated discs, across here, across here, across here, that you're only going to see in cardiac muscle. That didn't show up on the gold chloride stain, but when you hit it with a hematoxylin, it just pops out at you. So that's this idea of selecting the stain for the tissue type that I'm trying to get them to do to build up their own image bank. Just another example. The next slide should be coming up is mouse cerebrum. On your left, a typical slide that, again, you can take the tissues, we embed it, thin section it, put it under the microscope, stain it, and with a LAD multi-stain is something I've used for years because it's pretty good. It highlights different tissues based on density. But you know, from this distance, that's only at 100x, that could be kidney tissue for all we know. When you hit it with an appropriate stain, in this case the silver-based Golgi stain, you can truly see the pyramidal cells, and you can make a distinction between what is actually the neuronal transmission tissue and what is the bulk of it in terms of all the different glial cells. So it's the same slide, the same method, but just an alternate staining technique that bespeaks more of its function. Jumping back to last year's meeting, when those of you were at the New Orleans meeting when we did an IVF and we generated the sea urchin embryos, we couldn't take it more than about the 4-cell stage because time ran out in the workshop. But I do this routinely, and this is a much later stage of gastrulation. This is a little cheating here a little bit. This is a starfish, not a sea urchin, but the embryology is the same. And as you all know, there is zero depth of field under a microscope, and this was shot at 400x, so there's just no depth of field, and you have to make a value call. What are you going to highlight? The dermal denticles that eventually are going to characterize the echinoderm, or are you going to look at the gut tube, or are you going to look at the migration of the mesenchyme? You have to basically make that decision when you take that shot. I think it was Gabe's presentation last year or the year before that turned me on to Z-stacking. And I don't have the little fancy system, but we had an old Sorval microtome that was useless because it would vibrate every time you turned the handle. But the rocker arms still work and the advancements still work. So we took basically the thing apart and assembled the rocker arm onto the fine focus of the microscope. And now when you turn the crank on the microtome, you can focus down through the specimen at something like about maybe 30 to 40 micron. So that's basically your value call. Again, what do you want to see? There's, again, on your left, just looking at the argenteron, what eventually is going to form the gut. And this is a Z-stack with that really crude technique. You know, biology on a shoestring is kind of what I'm left with most of the time. So in this case, you can see clearly the argenteron. You can see the migration of the mesenchyme right and left that's going to make the mesodermal layer, and you can see the three-dimensionality of the exoskeleton already forming. So it's, for me as an embryologist, that's a much, much better image. Other thing we're playing with is as we're coming to seahorses, trust me. I work on seahorses, and I don't know much about things other than fishes. But the fish gut in general is not specified for regional absorption. It's not like our ileum versus jejunum. It's generally a absorptive tube. Not so the case in some of the more modern fishes, and seahorses are very modern fish. So we have a project going on to figure out, is the gut tube a great big absorptive tube because it's all microvilli, or is there regional specialization? Well, an easy way to do that is to take a material such as a low molecular weight protein or a sugar and then label it with something and then basically dissect out the gut and see where it has been endocytosed. So I have a student working on that right now, culturing yeast. Yeast feeds sugar, so we have Lucifer yellow dextran, the glucose is labeled with Lucifer yellow, feed it to the artemia, and this is just a dark field image, but then feed the artemia to the seahorses and then wait, dissect out the gut, and again, you'll get these great big endocytotic vessels. If it's uniform along the tube, answers that question. If it's pocketed along the tube, it answers that particular question, and then we'll follow it up with a labeled protein. But again, just different techniques that you can apply to your microscopy to solve that question, all very doable in a shoestring budget. One of my favorites is, I like osteology very much, and this is a technique that I'm sure you've seen used many a time, but clearing and staining of an embryo is something that I like the students to do. I can hand them a jar that has a clear and stained cat skeleton and explain to them about epiphyses and diaphyses and cartilage becoming eventually bone, but it's not quite the same as having them do that as a project. So we have a gross anatomy lab. Usually we're working with pregnant cats. Take the kittens out of the uterus and prepare them accordingly. They need to be skinned, which is a little bit gruesome. You know, skinning kittens is, you know, you get a bad rap for that, but then you put them in potassium hydroxide and it macerates the tissues, soak them in this solution of alizarin red S, also in the potassium hydroxide, and it bonds only where there is calcified layers. You, from there, put it into progressively stronger changes of glycerin, and it leaches out everything that isn't red. And where you see the red, of course, is the calcium. And then we can literally focus in and talk about calcification of the skeleton and problems that go on, why you don't involve, you know, full compact sports in children under 14 based on all this. It really fits nicely into the curriculum. So there's just some close-ups of that particular mouse arm. You're seeing the calcification here of the epiphysis and here in the diaphysis. I'm not really sure why the nails are going on, but I promise you I didn't paint those nails. It looks like they did. But again, you can see the calcification. That's, again, only where there is alizarin red here. And note on these particular bones, you have heavy calcification here and here, but there's a lot of cartilage in between. This, of course, is still developing bone, so a calcified sheath here on the diaphysis, calcification at the apophysis, but cartilage here and a lot of the cartilage here. That lends itself up for a counterstaining method that I like to use now that is, I think, doesn't work as clear as the alizarin, but it works really, really well. Well, I guess I have that coming up in a couple of slides, but I'll show you one where we can counterstain and bring out the cartilage as opposed to the calcified bone. So there is 100x and 400x compact bone drilling right through the epiphysis, or the diaphysis for that matter, the little Haversian canals or Volkmann canals, whichever way this is running, through which there would be a blood supply. And then it lays down this complete osteon with the osteocytes here along the caniculi, filling in the trabeculae, and that's a nice picture of bone. But if I want to talk to the students about, for example, osteoporosis, and what are we going to do about the lack of calcification, that really doesn't show much. It's just sort of a static picture. If you take the same tissue and then stain it with alizarin red, now you can see where the calcium is being deposited in those lamellae, and that's literally what gives compact bone its axial strength. If I did this same thing with osteoporotic bone, or maybe even in embryos that were culturing under a low calcium condition, hit it with alizarin red, you'd see these great big deficits where there should be, again, counterstained calcium. So very cheap, very easy to do. Within about a 3-hour lab, students can get these kind of results, and then it's up to them, again, to build up their own image bank. This is the one I was telling you about, another kitten, cleared and stained, first stained with alizarin red, and then you delete that with, of course, the glycerin, and then counterstain with Alcian blue, which does just the reverse. It ignores calcified tissue, but it settles in cartilage. And it's not really easy to see down here on the limbs, but if you put it under higher magnification here, this, of course, is all calcified with alizarin red, showing the deposition of the osteocytes. But this is the area that is still chondrocytes and all cartilage bone. You can probably see the big space between that chondrocyte and this chondrocyte that you wouldn't see in the ossified bone. And this is just a really good picture that I think, after the students have done the technique and they know what they're expecting and they get these results, they truly understand this process of chondrification and the developing of the endoskeleton and then progressive osteogenesis that results in basically just these long fused bones with cartilage only at the joints. So again, that's just all student imaging that you're seeing there. Okay. Getting back to the concept of choosing the stain, for example, the Golgi stain that brought out the boutons of the neuromuscular junction, I was going to take some trout embryos and walk you through this with a different class. This would be comparative anatomy. So there's a trout embryo that probably looks familiar. This is a brown trout. The ones I catch are much bigger than that, but not much. But this would be in transverse section, so literally an entire mirror image through the central axis, and then I'll show you some slides that are taken either through the eye or basically through the foregut, so you'll be able to see, again, neural tube and all that in position. So again, the assignment, I give the students the embryos, they have to take it from there, and what we're looking at would be something like this. For your orientation, this would be transverse, and what you're looking at here, this is the notochord. So the neural tube would be up here. This is the notochord, and that is their very first skeletal element, as I'm sure you know. These great big vacuolated cells that are wrapped by collagen, and that is the original attachment for the muscles. And then we can get into resorption of the notochord, chondrification of the skeleton, osteogenesis of the skeleton, but it starts with this. This is unstained, so you're just looking at the coloration of the tissues of the trout, maybe a little bit of light involved there. This is a stain with methylene blue, and then this, of course, is toluidine blue. Methylene blue is quick and easy and dirty, and you can make it with an alcohol base or a water base, but I always use water base in the lab. And I guess the nice thing about it is time is not really an issue. You can leave it on there for 30 seconds. You can leave it on there for 3 seconds. When you rinse it off, you pretty much get the same intensity. And it stains the tissues, but it doesn't stain the media that is around there, the embedding media. But again, all it does is bond to protein lipid, and everything is protein lipid, and it really doesn't make a distinction based on the density of the protein or the thickness of the lipid layer. It just shows you what is there. So I can point out to them notochord. Yes, you found the notochord. You also have muscles here, but it doesn't really show you much. Toluidine blue is an alcohol-based stain, and their time does work for or against you, because the longer you leave it on your thin section, it will permeate deeper and deeper and deeper into the tissues, and it also bonds more deeply to dense tissues such as protein compared to looser tissues like adipose tissue or the vacuolated cells of the notochord. So it gives you some differentiation between tissues, not just collective lipid protein, but maybe some distinction between, well, this is more lipid, this is more protein. And the other thing about it is, again, if you leave it on there longer to get a deeper stain, the downside is that it will stain, of course, all the media. So there's some, the JB4 resin media up there that would be a deep purple as well. But since it is alcohol stain, then you can wash it with alcohol as opposed to water and eliminate everything that isn't bonded onto some sort of tissue. So that's really where this came from. I much prefer it as an image. Again, notochord here, notochord here, but you can see the distinction of this blue layer here, which is actually ectodermal, and I'll come back to that one, and this layer on the outside that is actually the collagen mesodermal layer. So you're getting some differentiation between tissue types here, but this is really dramatic. This is muscle, and it's not specific for muscle, but this is cartilage. This is the hyaline cartilage that is basically making the original framework for the gill lattice. And you can clearly then distinguish between tissue types based on that simple choice of stain. Exact same fish, probably the same block. I'm sure it was the same block, maybe 3 or 4 sections deeper, just simply stained differently gives you a much better image, gives you more visual data. And then I started playing with Giemsa stain. We do a lot of work in embryology now. We do a lot of work with developmental genetics. So as genes are turned on, turned off, and it results in DNA transcription and eventually protein translation, you need to be able to see which cells are doing that at any phase of development. So Giemsa is wonderful for that. It bonds onto the phosphate part of the DNA molecule wherever you're making the adenine-thymine bond, and I can ignore the rest because as long as I can see something, I know those nuclei at the time are being either translated or replicated, but it lets me know what cells are active in the process. So I've used that for years in embryology, but we started trying it on these particular fixed specimens as well. It really doesn't show you much, and it's not supposed to show you much if you look at it in a cross-section like this. That actually has a little less detail than the toluidine blue, but it's a lot better than methylene blue. But then I started playing with the May-Grunwald stain, which is really, I guess, my favorite because it bonds to tissues, again, that are lipid protein based, but it is also selective for the pH, if you will, of the cytoplasm. So you have, acidophilic cells, and you have basophilic cells, and you have neutrophils. It was really developed for blood work, but it works really well in these tissues. So now you have a really clear distinction between what is muscle tissue, okay, what is cartilage, again, what is an ectodermal and a mesodermal layer, and I'm not really sure what's going on here, but I think that's probably just, it could be, as we call a fixation artifact, which means we don't know, or it could be a basal lamina. But again, a lot more visual data. So I got the idea, it's not my idea, I had read about this years ago, and then I thought, well, let's try it. Why not combine the two in a multi-stain and then see what happens? In theory, if you do this right, you're supposed to be able to see, you have an array, a rainbow of colors that if you understand what they are, they're telling you what particular tissue layer is there. Still kind of a work in progress, but let me show you what most recently one of the students did that I was very pleased with. This was a mouse embryo. We had cages of mice, and when we put the male cage next to the female cage, they both get the message, and the female will ovulate. So we sacrificed one of the ovaries, and you're looking at it, of course, in low mag here. This is about 40x. That'll show the distinction between the full Graafian follicle, maybe a post-ovulatory follicle that is now the corpus luteum, and then some little developing follicles here. And it's bringing it up on higher magnification, but this gave me much better resolution, much better distinction, visual data than any of the other individual stains. So this is the oocyte as it develops. You can see the zona pellucida clearly around there, this follicle cell layer that is, again, dividing very, very rapidly, shows up, I think, because of the Giemsa stain. The additional follicle cell layer that's going to be all around the outside that is making the estrogen, also dividing very, very rapidly. So I think the Giemsa really brings that out. But as far as the pH of the internal tissues, there's a big difference between a developing oocyte, which should be neutrophilic, compared to something like a Cortius luteum, which is going to be more of an acidic nature of cell. So it shows all that quite well. These are the developing oogonia before they even enter the process. And again, you can see a distinction between the nucleoplasm here, and I think that's all due to the May-Grunwald state. So I like that multi-stain, and I'm going to experiment with it a lot more as years go on. Just have to basically get better at it and figure out what hue is indicating what particular tissue type. Okay. Now, before I go into this one in cross-section, so again, you saw the trout earlier. This is just a close-up view. This will be sectioned right through the yolk reserve, the yolk sac here, through the foregut, up through the neural tube, and then cross-section will look like this. So here is the gut tube. This would be yolk. I'm not really concerned with that. We will be in the Seahorse pictures. The developing notochord that we already talked about, and the neural tube, just for your orientation. That doesn't really show a great deal of distinction. Okay. So this is a multi-stained embryo, but I stained the embryos whole mounts, and then embedded them and sectioned them. And the idea is to get the stain deeply into the tissues and see this differential, the distinction between, again, things that are an ectodermal and a mesodermal layer. Therein, I usually like to stop, and I don't want to insult anybody's intelligence here, because you've seen things that I don't want to see, believe me, from some of the slides. But I usually walk my students through a visualization exercise to remind them where this comes from and the importance of genes in this entire process, and therefore that's why we're looking at a Giemsa stain for the DNA action, and that's why we're looking at something like a May-Grunwald for different types of tissue based on the nature of the cytoplasm. I happen to start with a ping pong ball, and I say consider that an ovum. Put a little dot on there, and we'll consider that fertilization. And through repeated cleavages, 1 to 2, 2 to 4, where we have replication of the nuclei, more and more cells with fewer and fewer, more and more nuclei with fewer and fewer volumes of cytoplasm, you end up with something that kind of looks like a golf ball. And I say, all right, humor me that they're the same size, and make it a hollow golf ball. And all those little dimples on the outside that I think they have a name are cells that are genetically identical. And everybody says, yeah, and they yawn and web surf. I say, well, answer this question. How do you get nose and toes and bones and spleen and heart and everything in between that is so incredibly different in terms of its expression when they're all genetically identical? And they can't answer that. That's where gastrulation enters the picture. So I say press your thumb on there and make an indentation, and you've taken genetically identical cells, and now they're in different environments. That's the keys to gene expression. The cytoplasm will change based on the external environment. That changes the nucleoplasm, and that's differential gene expression. And they start to get the reason for this. I don't know if Lewis Wolpert, maybe many of you have heard of Lewis Wolpert. He's sort of the grand poobah in embryology. He's a Brit, about 89 years old. He has a quote he has a lot of quotes, but my favorite is that, I'll paraphrase something like, it is not birth, marriage, or death that is the most important event of your life, but gastrulation. And I think he's actually true of that, because if you survive the period of gastrulation, you have all the blocks necessary to make all the organ systems. So those blocks I've been talking about were ectoderm on the outside that'll make things like the entire nervous system, make most of the eye, all of the ear, pigmentation of the skin, the endoderm layer that basically makes the gut tube, and then the mesoderm layer that makes everything else, red organs, including, for example, the notochord that we were looking at. So if I have a differential stain that'll pick out mesodermal derivatives versus ectodermal derivatives versus endodermal derivatives, it's a really powerful tool to use during embryology. We can culture zebrafishes or medacas and fix them at various stages of development, multi-stain them, section them, and they can literally see the fate of these cells at a very, again, low-cost version. So this was the pre-embedded tissue. Again, the entire embryo is multi-stained, the Gimsa Grunwald, and it shows some things, but it doesn't show a great deal. So you have neural tube here, which doesn't really look that distinct, in my opinion, from the notochord. There's a little bit of red here that would be mesodermal, but not a great distinction. So this didn't work as well as I had hoped. But if I do pre-stain them and then embed them and section them, and then we take those sections on the glass slide and then restain them with the multi-stain with the May-Grunwald, it looks much better. This is the same, again, maybe 3 or 4 sections in, taken at about 50 micron. Now you're starting to see a big distinction between, this is muscle, which is all mesodermally derived. There's the notochord, which is mesodermally derived. This is cartilage, which is a combination of mesodermally derived tissue here, and it's beginning to ossify, I think, here. There's the neural tube that is entirely ectodermal, and based on the idea that you have Gimsa in there, you're picking up the nuclei in the cells of the neural tube versus basically what is all going to be the axons of the gray matter. So it really shows a lot compared to the previous slide. And this is just basically a higher magnification of the same thing. Okay. And then moving into the eye, I've been fascinated by the development of the eye. It was one of the first things I think I ever worked on, and I always work on fishes. So this would be our section basically through the head of the fish. And again, this is a pre-embedded, multi-stained embryo looking for the retina of the brown trout. So again, I have to precede this to my students with a lecture on how the eye forms, and yes, it is largely ectodermal, but there's a mesodermal component. When the brain is forming, and of course we have something like the diencephalon eventually forms, that's entirely ectodermal. The epidermis on the outside of the head is entirely ectodermal. When those two tissue layers physically bump, something just magical happens that I never get a kick out of seeing. The inner layer will buckle and fold a double-walled cup, and that is going to be what you're seeing here. This is the double-walled cup of the eye that eventually will form the retina, sensory retina, and the choroid layer. And all that stuff in blue is what you're seeing as an ectodermally derived result of that. The head of the skin on the outside surface of the head would then form the retina of the eye. The lens of the eye. But in terms of the retina, the tapetum layer, that basically optic cup is all ectodermally derived. And this little area here, this choroid fissure, turns out to be critically important because as the eye is expanding, as the diencephalon of the brain is expanding to bump into the head and then tuck and fold and make that cup, it has to physically touch. There's all these experiments that are rather grisly but eloquent where if you prevent those cells from physically touching, it's not just enough to be juxtaposed, they have to physically touch, you'll end up with no formation of an eye, so there would be just non-differentiated skin going from maybe forehead to cheek. B has to push that mesoderm out of the way for those two ectodermal layers to touch. But the mesoderm has to get back into the eye because basically that's what inflates it and gives it its ovoid shape. And it can be underinflated or overinflated and result in myopia or presbyopia, but that's all mesoderm. So where that flows back into the eye is this area of the choroid fissure. And what you're seeing here in this kind of reddish or purplish hue is the vitreous humor that is a mesodermal derivative. And without that, again, this would be an unflated eye, and you would see simply strict blue. So that's where it was. The pre-embedded one showed you the same thing, but pre-staining, embedding, post-staining shows it much, much better, I think. And that's why I really like this. The tapetum layer is its own, pigmented layer. That's melanocyte as well. The sensory retina here, but again, you're getting an idea of the importance of mesoderm in the development of the eye and in the development of the proper shape of the eye for vision. All right, so that's the differential staining that I'm playing with my students. And the whole idea, again, I'm in a teaching institution, the whole idea is to cement those concepts into their brain, and nobody has an attention span to listen to 50 minutes. I'll finish soon. So if I can briefly give them just a description and then put them to work, my evaluations go way up. Divorcing myself from my students that I love dearly and getting back to what I truly like to do. When Jack was talking about his images and he said, you know, something like love the place where you're shooting or develop a connection with the place, I can't say that I have that living where I live now, but I do have a connection with these beautiful animals. I've been working on seahorses now for quite some time. I can't harm them anymore, so I try to come up with all these non-invasive methods of working with them, and the best I've been able to do is get into museum specimens that are way past caring, and then look at microscopic techniques that I can use to work on these animals to answer questions without cutting their heads off anymore and things like that. So that's what this is all about, and this gets into correlative microscopy. I'll tell you a little about what I'm doing. These images will be monochrome for a couple of reasons, but this is the guy that I work on. I assume everybody knows that seahorse males give birth, but you never know. This is the swollen pouch, and this one's actually ready to start delivering. You can tell they settle down to the bottom of the tank, they grab onto something. And I now understand why it's called labor. You can see this thing pulsating and pulsating until eventually it'll crown with an embryo. So I know when they're going to happen. You can set up, you know, your camera for time lapse and maybe get some of those images. Getting this, I don't want to hurt that little guy. It's about to give birth, for heaven's sake. I don't want to cut the head off. So go into the museum, and I can find, you know, museum pickled specimens that might go back to the 1920s that have a pouch distended like that, cut open a little flap of pouch, take a little flap of the tissue, and start working on it with electron microscopy, which is mostly what I do. So that's what you're seeing here. If you were to resect this pouch, inside there, it kind of looks like an egg carton, and these are empty, obviously. But if you were to zoom in here, these are full of microvilli, which of course is for exchange, and they're seeing a couple of empty chambers here and some here that are still within their external egg envelope. And that basically is where we begin, because seahorses, as it turns out, hatch precociously. The gestation of this is a little guy. They don't call this the dwarf seahorse for nothing. The entire animal from crown to tail is about 1.5 centimeters. So they're little buggers. But nevertheless, they hatch precociously, so it takes about, from fertilization in the pouch to hatching, about 21 days or so, and about half of that is spent in these brood chambers, and then the other half is when they stay in the brood chambers, but they're hatched, so you would actually see a little coiled up version of a seahorse in here, and that's when I began my work answering this particular question. Oviparous, of course, means all nutrition is from the egg. Viviparous, they're receiving some sort of supplemental transfer, like a placenta. And most people think that it is this, and I beg to differ, because I've yet to find any true nutrient transfer. I find a lot of materials transfer, but not nutrient transfer. And I can tell you how we figure that out sometime. But nevertheless, this is where we begin. If you look at this picture, you know, again, there should be a seahorse here, one here, and these are still in the egg envelope. Going into the museum specimens, this is basically just a real quick and dirty Carnegie stages of seahorses. So these would all still be enveloped in the egg envelope and sitting there in the little wrapper, and these would be free inside those little pouches. So I was going to compare this stage to this stage. This is the hatching stage, so it's no longer an embryo, and I no longer care about them. But I'm going to compare this stage and this stage. And this is basically where I started to see a weight gain during development. We have a weight gain that would suggest you have transfer like across a placenta, but it isn't the case. The weight turns out to be calcium, which is making this spiny exoskeleton, and I'll show you those images. And then basically this is also the stage where they begin to synthesize hemoglobin. If you were to look at the vessel system here, they're clear, there's no cells in there. These have pinkish cells, and they begin the synthesis of hemoglobin, which, as you know, needs iron. So we're going to follow this for where does the iron come from for the hemoglobin, where does the calcium come from to make the skeleton. And this gets into more light microscopy, eventually EM. There's a cross-section of one of those embryos, again, neural tube, notochord. This is the gut tube, and here and here and here, these are monochrome images because these are, Gulf and Caribbean Research publishes your image is free if they're monochrome. They charge up the wazoo if they're color. But this was a Prussian blue test. So Prussian blue is an old-time histology stain that bonds to iron and only iron. And when you hit it with a light, it is, who'd have thunk it, right? Blue. And that indicates iron deposits. So this is a light micrograph of the Prussian blue, and these would be blue. There it is under higher magnification, and sure enough, those blue deposits are sitting right along the vessels lining the hindgut, which to me says, well, something's getting into the hindgut through the vessels, and it's carrying iron. And there's, again, Prussian blue, you can see the cells inside the vessels. So where does the iron come from? Those vessels under TEM show electron dense in the early stage, just before it begins to synthesize hemoglobin. When it's a hatchling, those electron dense, or those inclusions that used to be electron dense are completely vacant. And this is the stage when the synthesis begins and when the hemoglobin begins to appear in the embryo and disappears and appears along the hindgut. So I've got this Prussian blue that says there's iron there and it's disappearing. I've got TEM that says it was electron dense here. That's probably where the Prussian blue is reacting, and it's not here. But where does the correlative microscopy come in? That's EDAX, or X-ray diffraction, microanalysis. Basically, any element has its own characteristic X-ray curve, emission spectrum. You hit it with an X-ray beam, and boom, it sends up a peak. It doesn't distinguish between valence. It doesn't distinguish between things like ferrous iron and ferric iron, but it does distinguish between iron and everything else. So those inclusions here, okay, show a distinctive 6.39 iron peak. That could only be iron, and that backs up visual data of TEM with X-ray diffraction with Prussian blue. And the same thing down here, those same inclusions, you lose the iron peak. And that's, again, the iron story. Okay. As far as calcium, beginning the eruption of the dermal skeleton here on that later stage, yet still in the pouch embryo, and that in term, you see this big distinction between these parts of the dermal skeleton and eventually the eruption of these little crowns. That's their only defense mechanism, is this calcification of spines. I knew there were calcium, because, again, calcium carbonate, but the pouch, as it turns out, concentrates that seawater, concentrates the calcium, and then it gets stored in the brood pouch of the adult male, and eventually from the brood pouch of the adult male gets leached into the tissues. And this is, again, later stage, when most of that calcium has disappeared from the brood pouch of the male, but it shows up in deeper calcification, this actually being one of these scutes inside. So there again, a different histological stain, von Casa silver nitrate, where you basically, just like developing film, shows these black deposits only where there is calcium, and everything else turns out bleached white. So I've got, calcium showing up here and here, here, and then I'm assuming this is the same structure. We can then take that, the same thing, and put it under the X-ray diffraction analysis beam under the TEM, and you get a calcium spike. Lower here at the beginnings of the calcification, right around 3.79, and peaked here right at partition. So again, just more visual data combining light microscopy, TEM, and in this case, X-ray diffraction analysis. And with one exception, that'll end things. This ought to look familiar. When something works for me, I stick with it a lot. So, but this was the beginning, and I figured you've all seen so much and done so much. I was going to give you one more thing you could put on your curriculum vitae that you probably haven't seen before. Even if you've worked in proctology, how many of you have used a microscope to look up the anus of a starfish embryo? So that's the end of today's talk.